started. Okay, so this lab is all about gymnosperms. This is personally one of my favorite group of plants because I study two pine species that are native here to California, so I'm a little bit biased towards gymnosperms. But a quick review of basal land plants. So just a reminder that the basal land plants include the non-vascular land plants, so those were the liverworts and mosses, and then also the seedless vascular plants. So that included the two phyla, Lycopodiophyta and Manilophyta. So those were your leptosporangiate ferns, Selaginella, lycopods, etc. Those are all collectively known as the basal land plants. So for these guys, their spores were used for dispersal. So in order for the alternation of generations or for their life cycle to complete, those spores were essentially dormant until they were able to land in water and allowed them to germinate and produce a new gametophyte. So basically a brand new plant. So with basal land plants, gametangia were produced. So in this case, gametangia were the archegonia, which was the female reproductive structure that housed the egg. And then the antheridia was the male reproductive structure that housed the sperm. Now the egg and the sperm essentially came together, they fertilized and then produced an embryo, but none of that can happen until the spore lands in water. So if it's not in water or if it's not in some kind of really moist and wet environment, this life cycle essentially cannot complete. So for basal land plants, they required water and moist habitats in order for the free-living gametophyte to grow and reproduce. So that was specific to, once again, non-vascular plants and seedless vascular plants. I see a cat. So this is an example of what it possibly looked like millions of years ago. With a large body of water, it looks like we might have some moss growing, maybe some liverworts over here. But once again, these basal land plants really needed water and some moist habitat in order for their spores to be dispersed. This is another photo of what it possibly looked like during the Ordovician. We have a small body of water here. Looks like we have some maybe lycopods, I'm not exactly sure. It looks like we have a little bit of Selaginella here, but essentially with seedless vascular plants and non-vascular land plants, they also really didn't get that big. There were some members in the Lycopodiophyta, like Lepidodendron, which we didn't really talk about because those are extinct now, but those can get really, really big without any secondary xylem. But for the most part, non-vascular plants and seedless vascular plants are pretty small. And once again, they required water in order for their spores to disperse. So this is the phylogenetic tree with the basal land plants all the way to the left over here, and then the gymnosperms and the angiosperms all the way to the right, because these are the more derived groups. So these are the groups that evolved later on. We're specifically going to be focusing on the naked seed plants or the gymnosperms. But if we look over here to the left of the phylogenetic tree, we have Hepatophyta, also known as Marchantiophyta, which is the liverworts. If you see the term Hepatophyta, that's just the old name for the liverworts. We call them Marchantiophyta now, but it's referring to the same exact group. So again, that's the liverworts. And then over here we have Bryophyta, so the mosses. So these are the non-vascular land plants. And then going into the seedless vascular land plants over here, we have Lycopodiophyta. So this is the phylum that included the lycopods and Selaginella. And then we have Pteridiophyta. This is another old name, but it is referring to the Manilophytes or Manilophyta. So that includes your leptosporangiate ferns or your true ferns, aquatic ferns like Azolla, and then also the horsetails and the whisk ferns. But all of these together make up the basal land plants. Now, in between the evolution of basal land plants and the seed plants, so that includes gymnosperms and angiosperms, there were four really important characteristics that evolved that essentially allowed these seed plants to really dominate and succeed. So one of them is the evolution of seeds. Now, with the evolution of seeds, it essentially made spores obsolete because with the evolution of seeds and actually pollen as well, they no longer needed water or that moist environment to complete that life cycle. So now these plants can live in a lot drier habitats. So that was the evolution of seeds and pollen. They happened at around the same time. And then we also have the evolution of secondary xylem and phloem. Now keep in mind that the seedless vascular plants, so Lycopodiophyta and Manilophyta, they do have true vascular tissue, so a true xylem and phloem, but they are not secondary xylem and phloem, so they don't have true wood or bark. So secondary xylem and phloem was really a characteristic of the seed plants. And then now we also have the gametophyte being housed on the sporophyte. So all the plants that we're going to be looking at from now on are sporophyte dominant. Keep in mind that seedless vascular plants were also sporophyte dominant, but it still relied on the gametophyte during its early stages. But now the gametophyte is completely reliant on the sporophyte. It's a lot more reduced now at this point as well. Any questions so far? Thumbs up if we're good. Okay, cool. Okay, so like I said, what I have circled right here in red is what we're going to be focusing on. We're going to be talking about the 3 extant or living phyla of the gymnosperms. So that is Psychedophyta, which is the most basal one, Ginkgophyta, and then Coniferophyta, which is most likely what you guys are more familiar with. Okay, so the gymnosperms are a monophyletic group, which means it includes its ancestor and all of its descendants. There were 4 developments in evolution that allowed gymnosperms to really dominate and become successful and pretty much outcompete seedless vascular plants and non-vascular plants. We just talked about these 4, but I'm going to go over it again pretty quickly. One of them is gametophytes being housed on the sporophyte. So once again, these plants are sporophyte dominant or diploid dominant. You'll notice that as you move on in evolutionary time, the gametophytes become more and more reduced and less complex, whereas the sporophytes really start to dominate. And then you also have the evolution of seed and pollen. This happened pretty much simultaneously. And then also the evolution of secondary xylem and phloem, so true wood and bark. And gymnosperms are often referred to as naked seed plants because they don't have any fruit. You don't see any true fruit until you get to the angiosperms. So although some of the cones of gymnosperms, like for example, ginkgos or podocarpus, and junipers, sometimes they look like they're an actual fruit because they have a fleshy outer layer or a fleshy seed coat, but they're actually just cones. So no real fruit in the gymnosperm, so basically no fleshy sporophyte tissue that surrounds and essentially protects the seed. In fruits, the fleshy sporophyte tissue is the part that you usually eat, but none of that in the gymnosperms. Okay, so all gymnosperms evolved from heterosporous plant ancestors. Heterosporous meaning that it'll produce 2 distinct spore types, 1 female spore and 1 male spore. If it's a homosporous species, like for example in lycopods or some non-vascular land plants, they'll usually produce 1 spore with the male and the female parts, so with the archegonia and the antheridia. But gymnosperms typically evolved from heterosporous ancestors. So once again, that is a separate female spore and a separate male spore. Now, anytime you see the prefix mega, that is referring to the female. And when you see the prefix micro, that is referring to the male. So whether that is a megaspore, megagametophyte, megasporangiate cone, it's just always referring to the female. And it's the same thing for the male. It's just that they throw micro in front. So all gymnosperms also will contain secondary tissues, so that is secondary xylem and phloem. Secondary xylem and phloem were very important because it allowed these plants to get much taller and grow better in comparison to basal land plants, and it also allowed them to resist bending and damage as well. So because they were able to grow a lot taller, they were able to disperse their seeds a lot farther away, excuse me, and they were also to compete better for light and resources. So as stated earlier, the gymnosperms are all sporophyte dominant or diploid dominant. So the gametophyte is basically completely reduced and is housed on the sporophyte. Typically, the gametophyte is going to be housed inside the strobili or the cones. So with the plants being sporophyte dominant, it allowed them to survive in much drier conditions because they no longer need water for transport of their gametes or the establishment of gametophytes, so the archegonia or the antheridia. So essentially, with gymnosperms and even angiosperms as well, spores were no longer used for dispersal, so they essentially became obsolete, I like to think, because now they're using seeds and pollen for dispersal. So once again, they no longer depend on water, so they can disperse more inland and live at higher elevations, and they can live in drier, more arid habitats as well because of this. Okay, any questions before I go into the male cone?
With the more basal plants,
Mhm.
wasn't it like before, wasn't the sporophyte housed on the gametophyte?
Yeah.
So that's reversed now?
Yeah, so it's completely reversed now. So, but keep in mind with seedless vascular plants, so that was like the manilophytes and the lycopods, they were sporophyte dominant, but the sporophyte was still housed on the gametophyte and still relied on the gametophyte, at least during the really early stages of its life cycle. So now it's completely switched where the sporophyte is dominant and the gametophyte is now housed within the sporophyte.
Okay, sounds good.
Usually in the clones too. Okay, good question. Okay, so now I'm going to go first into the male or the microsporangiate cones. Okay, we have a question. Okay, so what I mean by longer lived is that they have a longer life cycle. So they can usually live a lot longer compared to seedless vascular plants or non-vascular plants. Like if you look at some gymnosperms, like for example, redwoods in the conifer phyla or some pine species, they can live up to hundreds, even thousands of years. I think the oldest pine is the bristlecone pine, and I think from what I remember, it's about 5,000 years. So pretty much they can live a lot longer. They have a lot longer life cycle compared to seedless vascular and non-vascular plants, especially because they no longer need water as well to disperse at least. Okay, good questions. Okay, so now I'm going to go into the male cone. So just keep in mind that anytime you see the prefix micro, that is referring to the male, and when you see the prefix mega, that is referring to the female. So the male strobili or the microsporangiate cones is what I have here in the image on the right. This is what the anatomy looks like essentially. So the microsporangia, which are these large sacs that my cursor is kind of going around, this is where meiosis is going to take place and house the haploid microspores. Now, the microsporangia are housed on microsporophylls, which are essentially the scales of the cone. So anytime you see the word phyll, that is referring to the leaf. So for example, like phyllotaxy refers to leaf arrangement. Megaphyll refers to large leaf. Sporophyll is essentially modified leaf that houses the sporangia, which is why it's called a sporophyll. And then when you just throw the word micro in front, that's referring to male. If it's megasporophyll, that's just the modified leaf of a female. So once again, the microsporophyll is essentially where my cursor is on. That's like this purplish area right here. Again, those are basically just the scales of the cone. And within the microsporophyll, it's going to house a microsporangium. So that's this sac-like structure right here that this bracket is pointing to and my cursor is kind of going around. And then inside the microsporangia is where meiosis is going to happen and where the male haploid microspores are going to be housed. Well, eventually those haploid microspores are going to germinate and they're going to produce your microgametophyte or your pollen grain basically. So all of that is going to take place in the microsporangium. So depending on what stage this cone is in, if you look at it under a microscope, they might still be haploid microspores, but for the most part, they'll probably be a pollen grain or a male gametophyte already. But that's all going to be within the microsporangia. Does that make sense? Okay, I have another picture here where it's kind of zoomed in. So once again, the microsporophyll, that's basically the scales of the cone. Once again, it's just a modified leaf that houses the microsporangium. So that's this sac-like structure right here that my cursor is kind of going around. So this is one microsporangium, and then this is another microsporangium. And then below it, it's kind of cut off, but that would be another microsporangium right there. Inside the microsporangium is where meiosis takes place. So these individual, like tiny little circles, that is all an individual haploid microspore. Eventually, those haploid microspores are going to germinate, and then they're going to become your pollen grains. And then when they do become pollen grains, the male cones will eventually open. Sometimes they'll really just crumble apart. That way the pollen can get dispersed in the wind. But once again, that's all housed within the microsporangium. So here's like your microsporophyll here. Here's another microsporophyll. And once again, it's just a modified leaf that houses the microsporangium. Does this make sense? The anatomy of it? Okay, cool. Okay, so Keep in mind that inside the microsporangia is where the microspores are, and then eventually those will germinate to become your pollen grain, also known as your male gametophyte. Now, your pollen grain is made up of two cells. You have your generative cell, which will produce your two sperm, so your two male gametes, and then your tube cell, which will generate the pollen tube. And I'll talk about what the pollen tube does in a second. But the pollen grain, it basically provides protection for the male gametes, and with the development of the pollen grain, since it's most likely going to be dispersed through wind now, it no longer needs water for dispersal. So remember that spores from seedless vascular plants and non-vascular plants, they needed water in order for that life cycle to be completed. With pollen grains, they're dispersed in the wind, so they no longer need water. Okay, so like I said, the pollen grain is made up of two cells. You have your tube cell, which will generate the pollen tube. What the pollen tube basically does when it does form is locates the micropyle, which is like this small little opening in the female ovule. When the pollen grain lands on the micropyle of the female ovule, once again, it's just a small little opening. Basically, what it'll do is that tube cell will get a cue to generate the pollen tube, and then that pollen tube will basically inject itself into the embryo sac. Well, it'll inject itself into the archegonia first, and then it'll burst, release the sperm will fertilize the egg, and then you'll get a brand new embryo. And those two sperm come from the generative cell. So the pollen tube is basically carrying that sperm that's produced from the generative cell to the egg. I'll show you guys what the pollination looks like in a second too. But just keep in mind, tube cell produces your pollen tube, generative cell produces your two sperm cells, and those are the two cells within one single pollen grain. Okay, so pollen tube, once again from the tube cell, I kind of just talked about this already, but it's pretty much digesting the path to the egg. So once that pollen grain gets dispersed in the wind, it'll hopefully land on the micropyle. So again, it's that small opening on the surface of the ovule, which is the female cone, part of the cone essentially. It'll enter the embryo sac in the egg, and then it'll burst and release the sperm. So here's a photo of the pollen tube right here, carrying the two sperm cells. That'll eventually inject itself into the archegonia, it'll burst, release the sperm, and then fertilize the egg. Now, even though it produces two sperm, there's actually usually two eggs as well in the archegonia. Usually only one of the eggs will get fertilized and the other sperm will die. Okay, so now we're going to go into the female strobili or the megasporangiate cones. It's the same exact terminology with the males, where with the male cones, you had your microsporangia housed on the microsporophyle. Same exact thing for the females, where you have your megasporangia housed on the megasporophylls. So if we look at the anatomy of a female cone right here, these ovuliferous scales that it's referring to in the picture, that's essentially the megasporophyll. So the modified leaf, or in this case, it's a scale that houses the megasporangia, which is essentially the ovule, which is down here at the bottom. So this entire structure is the ovule. If you were to zoom in on the ovule, which is right here on the right, the outermost layer would be the integument. That's essentially providing protection for the megaspore, which is eventually going to become your female gametophyte. And then this integument, which again, that's the outermost layer of the ovule, that'll eventually become your seed coat when fertilization occurs and you get an embryo. So right now, this is an unfertilized ovule, which is why this is still an integument. This is the small opening on the surface of the ovule, also known as the micropyle. So that's what the pollen grain is trying to find. And then to the inside of the integument, you have your megasporangium. So remember, that is the structure that houses your haploid megaspore. So this is where meiosis is going to take place. And then eventually that haploid megaspore will germinate and become your female gametophyte, which will produce your eggs essentially. So again, this entire structure is the ovule, and that's found at the very end of this scale right here, or the megasporophyll. So this is one ovule with the megasporophyll, another ovule right here on my cursor with the megasporophyll, another ovule with the megasporophyll, so on and so forth. And then this is just zoomed in on an individual ovule. Okay, So within the ovule, you have a diploid megaspore mother cell that's going to undergo meiosis. So that's within specifically the megasporangium of the ovule, where meiosis takes place, and it's going to produce 4 haploid megaspores. But usually only one of those megaspores survives. So if you look at this image over here on the top right, there's that one haploid megaspore that survived. So 4 gets produced from the diploid megaspore mother cell, but only one of them survives. And then that one megaspore that survives is gonna germinate and produce your megagametophyte or your female gametophyte, which you see in this image below that. So that is your archegonia with the 2 eggs. So like I said, from the pollen grain, the generative cell of that pollen grain will produce 2 sperm, just like how the megagametophyte or the archegonia produces 2 eggs, but typically only one of them is going to get fertilized. And all of that is enclosed within the ovule. So once again, on this image at the top right, here's your megasporaphyll or your ovuliferous scale. Here's your ovule down at the bottom. And then here's your integument with the micropyle, so that small opening that the pollen grain is trying to find. Here's your megasporangium with your haploid megaspore in the center. And then eventually your haploid megaspore is going to germinate, produce your female gametophyte, which is right here down below it with the archegonia and the 2 eggs. Okay, so now we're going to talk about pollination. I really like this image at the top right here. So here on the far left, we have our individual microsporangium. And remember, within the microsporangium, that's where meiosis takes place, and it's where the haploid microspores are going to be housed. But eventually, the haploid microspores are going to germinate and become individual pollen grains or male gametophytes. So that's what these little circles are. This is one pollen grain, another pollen grain, and then another pollen grain. So eventually, that male cone is going to crumble, it's going to break apart, so that these pollen grains can get dispersed in the wind. When it gets dispersed in the wind, it's flying, and then hopefully it'll land on a female ovule, but it's going to try to find the micropyle, so that small opening on the surface of the ovule, specifically the integument. So remember that the integument is the outermost layer of that ovule. When it does find the micropyle of that integument, that pollen grain, specifically the tube cell, is going to get a cue to produce your pollen tube, which you see on this image on the right here. As soon as it forms that pollen tube, it's going to basically carry that 2 sperm cells that's produced from the generative cell with it, so that it can fertilize the egg. Keep in mind, usually only one of the sperm is able to fertilize the egg, as one of them dies, so the other egg cannot get fertilized. So usually only one embryo or one zygote will form. Okay, so in this image right here, I basically just have a picture of what a closed cone looks like and an open cone. There are some cones that will not open unless they're exposed to really, really high temperatures. Sometimes it requires fire for these cones to open. Those are typically referred to as serotinous cones. Actually, one of the pine species that I study in California, which is Pinus tenuata, they're one of the 3 serotinous cones. But yeah, they won't open until they have a really hot temperature. So unfortunately, if they're not open, their seeds cannot get dispersed. If their seeds don't get dispersed, they basically can't produce a brand new gametophyte or a new plant. So, however, most of the cones that are native to California will open. There's only 3 that are technically closed cone pines, but those cones do need to open in order for their seeds to get dispersed.
Do they need to be open to get fertilized or just dispersed?
Just dispersed. Because with the pollen grain is essentially completing the fertilization. So that's from the male cones. Okay, so with the seed, when it does get mature, there are 3 different generations. 2 of them are diploid, 1 of them is haploid. One of the diploid generations is the seed coat. So remember that the seed coat was derived from the integument of the unfertilized female ovule. So that is the outermost layer that's essentially providing protection for the embryo, and it allows the embryo to go dormant until environmental conditions are favorable, and yeah, essentially provides protection for the embryo. So that's the seed coat. That is a diploid generation. And then to the inside of that seed coat, you have your female gametophyte. This essentially acts as the food supply for your diploid embryo. So this is where the embryo is taking all of its nutrients from the female gametophyte. So the female gametophyte is haploid, and then your embryo is diploid. So again, 3 different generations within the seed: your diploid embryo, your diploid seed coat, and then your haploid female gametophyte. Any questions on this so far? We're good? One in the chat, it looks like. Okay. Okay, so now I'm going to be going over each individual extant phyla within the naked seed plants. The first one is Psychodophyta. So these were dominant during the Mesozoic. They're often referred to as the early gymnosperm because they're the more basal one. These are, or they do have monopodial growth. So if you think back to seedless vascular plants and non-vascular plants, most of them had dichotomous branching. So that's the earliest form of branching. So we're now moving into plants that mostly have monopodial growth. There are still some species that retain that basal characteristic of dichotomous branching, but for the most part, they are monopodial, meaning that they have one axis or one main stem that doesn't branch, or it doesn't branch frequently at least. So Psychodophyta do have really woody stems with a wide pith. Although they do produce secondary xylem, the majority of their shoot is actually pith. So it's actually mainly parenchyma compared to xylem. So cycads are evergreen, meaning that they're going to keep their leaves on during the entire season. They're not going to drop it during the winter or the summer or whatever it is. They'll keep it on for the entire season. So because they're evergreen, they have really stiff pinnately compound leaves that contain a lot of lignin in them. So remember, lignin is that chemical that adds a lot of rigidity to the cell walls. And they need a lot of that lignin because they're evergreen. So they need to last a long time. And it also prevents damage to the tissues if they're dehydrated, and also helps prevent cavitation as well. So remember, that's essentially air bubbles within the individual xylem cells. So cycads do still have flagellated sperm in which their pollen is going to send out a haustorial tube that will burst when the sperm swim down to find the egg. So their sperm is still flagellated. And for the most part, cycads are dioecious, meaning that they have separate male and female plants. So they'll have a male plant with just male cones and then a female plant with just female cones. Okay, there are 4 genuses that we're going to be looking at within the Psychodophyta. We have Psychus, which is over here on the bottom, and then Zamia, Encephalartos, and Dioun. Now, Psychus, I feel like, is one of the easier ones to tell apart amongst the 4 because they have really thin leaflets that end in a spine. But not only that, if you look closely at Psychus, I know it's hard to see in this image, but there should be pretty good pictures on the slides PowerPoint that help you answer the questions. But if you look at the center of each individual leaflet, there's going to be an indentation going down the middle. And that indentation is the one single vein or one vascular bundle that each individual leaflet has. So that's how you can tell apart Psychus. Now Zamia, which is one of my favorites, also pretty easy to tell apart because they have smooth or rounded leaf edges, whereas the other 2, Encephalartos and Dioun, they have spines on their leaf edges. But Zamia is completely smooth on the edges of their leaflets. And when you touch Zamia too, compared to the other 3 genuses, the feeling of Zamia is pretty like fuzzy and pretty hairy. So if you ever get a chance to go to the Mesozoic garden again, once this quarantine is all over, you go ahead and touch Zamia and it'll feel like really fuzzy. But so these guys, once again, they have their really rounded leaf edges, whereas Encephalartos and Dioun both have spines on the edges of their leaflets. Sometimes it's pretty hard to tell the difference between Encephalartos and Dioun, but typically Dioun has more narrow leaflets, whereas Encephalartos is usually a lot wider and more broad. But both do have spines on the edges of their leaves, whereas Zamia is smooth on their leaflets, and then Psychus will have that one vascular bundle going down the center. Okay, so for the strobili or for the cones of cycads, both the male and female cones are honestly pretty large, but the female cones are much bigger and they're typically going to be in the center of the plant. So over here on the bottom that my cursor is on, this is one entire female cone. It's huge. Eventually, this female cone is going to break apart, so it's going to crumble apart, which is why we refer to the female cones of cycads as ephemeral. They're going to crumble apart to disperse these really nice, colorful seeds. So this is a female cone over here that my cursor is on that's already broken apart. And these individual red circle structures, these are the seeds. The male cones, which is right above it, are typically more woody and persistent, so they're not going to break apart as easily compared to the female cones. But the female cones have to break apart so that they can disperse these seeds. So this is a male cone right here, a more mature male cone on a cycad plant, and then here's another male cone in the center of that plant that's starting to form. But male cones are woody and persistent. Female cones are typically much larger because they house tons and tons of seeds, but they are ephemeral, meaning that they will crumble apart eventually. And sometimes female cones can also exude nectar to attract pollinators as well. But with cycads, they're actually mainly animal pollinated rather than wind pollinated. And the reason why is because cycads are mainly found in really dense tropical forests that have a lot of vegetation. So it's actually really hard to disperse their seeds and their pollen in the wind. So instead, they're going to be animal pollinated. The animals will usually take the seeds at a much further distance compared to the wind for these guys, which is also why the cones of cycads and even the seeds as well are really bright and colorful, and that's to just attract certain vertebrate animals. But this picture over here on the right is actually a picture I took. These are three female cones on I believe this is an encephalartis, but you can see that it hasn't opened yet, but you can see the Red Sea kind of peeking through right over here. So looks like it's about to open, but this is in the Mesozoic garden on campus. Okay, so now the next group that we're going to be looking at is Ginkgophyta. So within this phylum, there's only one species left in this entire phylum. Everything else pretty much went extinct. So the only species left is Ginkgo biloba, which is seen over here on the right. So similar to Cycadophyta, Ginkgophyta is dioecious, so they have separate male and female plants, and they also have flagellated sperm, just like Cycadophyta. But the only really main difference or big difference is that Ginkgophyta is deciduous, so they will drop their leaves during the wintertime. So if you go to the Mesozoic garden on campus and you see a couple of naked trees that don't have leaves, most likely that is the ginkgos. They should have leaves right now, but if you go around September, October, November, they're probably not going to have their leaves. But they are winter deciduous. That's typically to prevent freeze-induced cavitation, whereas cycads are evergreen. So that's the one big difference between the two phyla. So ginkgo leaves are typically fan-shaped. However, there are two different leaf morphologies depending on if they're a long shoot leaf or a short shoot leaves. So I'll show you guys what the long shoot and short shoots actually look like in the next picture. But long shoot leaves, so leaves that occur on the long shoots, will typically have this really nice deep lobing. So this nice kind of V in the very center of that leaf, which you see right here in these two images as well. They'll usually have that nice deep V in the very center of the leaf, whereas leaves that occur on these short shoots are usually going to be smooth at the top. So when they're smooth at the top like this, we usually call that entire. Once again, that just means that they don't have that nice deep lobing that these long shoot leaves have. So short shoot leaves are going to be pretty much, for the most part, smooth at the top, and long shoot leaves are going to have that nice deep lobing. Now for the cones of ginkgos, both the female cones or the male cones can be found where the short shoot leaves are found. So you can think of, for example, this image right here. The long shoot, I like to think of that as pretty much like the main shoot or the main stem. So this is the long shoot right here, and then these like lateral branches are basically the short shoots, but this image right here refers to it as a dwarf shoot, but it's basically the short shoot. And then right here, this would be your long shoot. where you would have the long shoot leaves that have the deeper lobing, so that V in the center. And then this is your short shoot right here. You can see some male cones forming, and then the short shoot leaves, which are more smooth up at the top. But female cones, just like the female cones of cycads, they also can release some nectar to attract pollinators as well. But for the most part, the female cones and the male cones are going to be found on the short shoot, so the dwarf shoots right here. Okay, so the seeds of ginkgo are often thought to be fruit because they have that really fleshy seed coat, that really fleshy outer layer, but keep in mind that they're not a fruit. Evolution of fruit does not happen until we get to the angiosperms, which is next week, so they are technically a strobili, I guess, but they do have 3 layers to their seed coat. They have 2 hard layers and 1 fleshy outer layer, and then the innermost region would be your haploid female gametophyte, and then your embryo to the inside of that, which is diploid. So once again, though, the seed coat is diploid, embryo is diploid, and then your female gametophyte, where the embryo is taking its nutrients from, is haploid. So even though it resembles a fruit, it is not a fruit. It just has a really nice fleshy outer layer. The seeds of the female plant, those smell very interesting. You guys kind of lucked out on that one, because usually we'll bring in the female seeds in a Ziploc bag, and we'll tell our students not to open it, but somebody always opens it and it ends up smelling. It smells pretty bad. I do also know that ginkgo biloba is actually also used as a supplement, I think it's digestion. I'm not 100% sure, but I know people use it as supplements for something. Okay, so now we're going to go over the Coniferophyta. This is most likely the phylum that you're most familiar with because it includes pines, redwoods, also some really interesting ones like Podocarpus and Araucaria that are native to the southern hemisphere, and then the very interesting and morphologically diverse Nitales. I'm going to talk about that last because those are really weird, but they're cool. So most species within this phylum are monoecious, meaning that they're going to have one plant with both the male and the female cones, so both the male and the female gametophyte on the same plant. Now, what I do know with a lot of pines is that typically they'll produce their male cones closer to the base of the tree, so lower down in the tree, and then the female cones are typically going to be higher up, and that's usually to prevent self-fertilization. It's not always true, but for the most part, male cones closer to the bottom, female cones at the top. So again, most of the members of the Coniferophyta are monoecious, but there are some southern hemisphere conifers that you guys will look at in the lab, like Podocarpus and Araucaria, that are dioecious. So they have a male plant and a female plant. And the Nitales are also dioecious as well for the most part. So the cones of conifers are completely switched with the cones of cycads. So with the cycads, the male and the female were pretty large, but the males were the ones for cycads that were woody and persistent. For the conifers, the males, which I have over here on the left, are very, very small and they are ephemeral. So the males in this case are the ones that are going to crumble apart eventually, and they have to crumble apart so that they can disperse their pollen, because their pollen is wind pollinated. Whereas the females, which I have an example over here on the right, are really large. They're typically lignified, and these are the cones that will last a long time as well. So I have a couple more examples of some male cones right here. Remember that they are smaller compared to the female cones. So I believe this one is from Pinus lambertiana, or the sugar pine. That one's native to California. And then I think these two are both Pinus sylvestris, but these are individual male cones that are very, very small. And again, they have to be small and ephemeral so that they can disperse their pollen in the wind. And then here are some examples of female cones. I have the coast redwood or Sequoia sempervirens here. Again, the female cones are going to be much larger compared to the male cones. There are some female cones that obviously are not as big as other female cones from different species, but in comparison to the male cones at least, they're much larger. Then we have Pinus lambertiana again, or the sugar pine, and then a shameless plug for my thesis. These are the two pine species that I study. This one's Pinus coulteri. Also known as the widowmaker, because these cones get huge and they're pretty heavy. And if you look at the scales closely, they kind of end in like a spine, almost like a talon. So they're called a widowmaker because supposedly if they fall on your head, you're either severely injured or just dead. But yeah, Pinus coulteri, and then right here on the bottom is Pinus attenuata. So these are one of the closed cone pines of California. Specifically, they're serotinous cones, so they won't open unless they're exposed to a fire or really, really hot temperatures. But okay, so those are the female and the male cones. So remember that the male cones are ephemeral so that they can eventually open and crumble apart and disperse their pollen in the wind. So the way that these individual pollen grains can kind of travel a lot farther and float in the air is through these bladders that they have at the ends of their pollen grain, also known, or what I like to call them, as Mickey Mouse ears. So these are the bladders right here that help them disperse in the wind. Over here on the left is a immature male gametophyte, so immature pollen grain. And then over here on the right, when that pollen grain has located that micropyle of the integument, so that unfertilized female ovule, that tube cell, which is right here, will get a cue to produce the pollen tube. And this pollen tube is what's going to basically inject itself into the embryo sac and fertilize the egg. But keep in mind that 2 sperm cells are produced from the generative cell, which is right here at the top. But usually 1 sperm will fertilize the egg and the other one will die. And because they have these bladders now that kind of help them float in the wind, they no longer have flagellated sperm. Flagellated sperm was specific to ginkgos and cycads, but they do require a pollen tube to reach the eggs. So that would be this structure right here that's produced from the tube cell. So again, immature male gametophyte, immature male gametophyte, and then a mature male gametophyte. So I think of it as like Mickey Mouse and a little baby elephant, and the trunk is their pollen tube. Okay, any questions so far before I move on to the last bit? You're okay? Okay. Okay, so now we are going to talk about the Nitales. So this is a group found within the conifers. Early evolutionary forms were lianas or vines that had narrow stems and reduced leaves, but the Nitales are very, very diverse in terms of their morphology. We have shrubs, vines, we have trees, pretty much everything within the Nitales, but they're were also thought to be, I believe, the closest sister lineage to the angiosperms. And the reason why is because they actually have vessels or vessel elements for water transport. Keep in mind with gymnosperms, for cycads, ginkgos, and most conifers, they utilize tracheids as their main cell for water transport. But the Nitales have vessels, which is a characteristic that is specific to angiosperms, which is why the Nitales were thought to be the closest sister lineage to angiosperms. Not only that, they also have double fertilization, which resulted in the production of 2 embryos. Even though one of them dies, both of them get fertilized. Another characteristic that is specific to the angiosperms, but it turns out when the DNA of the Nitales were analyzed, they were actually more closely related to the conifers and not the angiosperms at all. So, which means that these 2 characteristics, vessels and double fertilization, were a result of convergent evolution, meaning that they evolved very similar traits that have a similar function, but they don't have the same common ancestor. And they also have their male strobili, or their male cones, pretty much look like flowers as well, the way that they kind of conglomerate, but they're not angiosperms. They are conifers, meaning that they are a gymnosperm. So there are 3 different species that we're going to be looking at within the Nitales. We have Welwitschia. Welwitschia is native to the Namibian desert of Africa, but if you ever get a chance to go to the Huntington Garden when all of this is over, I believe they have a Welwitschia still there. These guys are really crazy. They only produce 2 leaves during its entire lifetime from their basal meristem, and their basal meristem can also produce wood as well, which is really weird. But And then we also have ephedra. Ephedra sometimes looks like equisitales or horsetails to me. Again, this group is really morphologically diverse, but we often use ephedra to look at the vessels, which everything in all members of the Nitales have, but they just look really nice than ephedra. But their cones are much smaller in comparison to Welwitschia. And then finally, we have Needham, which definitely looks like an angiosperm. Even their cones look like angiosperms are a fruit of an angiosperm because they have that really fleshy outer layer. But some pictures of some of these Nitales. Over here on the left, we have Welwitschia mirabilis. Remember that these only produce 2 leaves during its entire life cycle. So this would be 1 leaf right here, and then this is another leaf over here on the right. It looks like it could be multiple leaves because the leaves started to form some tears when it got older, but it's really only 2 leaves. So here's 1 leaf, here's a second leaf, and then here's the basal meristem in the center. And then we have ephedra nevadensis right over here, which I believe you're also going to be looking at your walk, or during the virtual walk at least. And then we have Needham. This is what the tree looks like, kind of zoomed out. But like I said, they really do look like an angiosperm, especially their cones, because they have that really nice fleshy outer layer or seed coat. But yeah, so that is it for the lecture. Once again, this PowerPoint should have been posted to your Blackboard already, but I did record this lecture, so I'm gonna go ahead and upload this to YouTube once this session is over, and then I'll send the link to Lauren so that you guys have access to it. But do you guys have any other questions? Do I need to go over anything again? No? Okay. Well, that's pretty much it on my end. Let me go ahead and stop recording.